I'm going to start with a review of transcription, which was the last thing that we looked at in lecture. And you can see transcription here is shown in the image. Remember the promoter region. The promoter region is where RNA polymerase is going to bind. You also see that leader sequence. The leader sequence is going to code for the Shine-Dalgarno sequence, which is in messenger RNA, and we're going to see the role of that in translation. And then you can see that the initiation codon, the start codon, comes next. So I'm going to show you this video. It's about 10 minutes long, but it explains this whole process. And then we're going to go to transcription termination. Transcription termination has two events that can occur in prokaryotes. We're going to look at a video then that compares the two, row independent and row dependent termination. So I'm going to take you to YouTube to watch this video.
This video will be on the process of transcription in prokaryotes. So there are three stages: initiation, or how things get started, how the RNA polymerase gets to the gene; elongation, how the RNA is produced; and then termination, how things end up, how the cell knows where to stop transcribing and where the end of the gene is. These processes are different in prokaryotes and eukaryotes, and so we will be talking separately about prokaryotes and eukaryotes. This particular video is just looking at prokaryotes. First, I'm going to look a little at the structure of a prokaryotic gene. This is a general gene structure. So every gene at the DNA level, there's a region that is going to be transcribed, right? So this is the RNA coding region in orange, and right before that there are some sequences called a promoter, which help that RNA polymerase to get recruited, and at the end there are some sequences called a terminator, which tell it to stop transcribing. Now every time a particular gene is transcribed, the same exact region is always transcribed. So it always starts at the same base, transcribing, and ends at the same place as well. If you look at this RNA coding sequence and then look down here at the RNA that gets made, what you might also notice is that the protein coding sequence is slightly different than the RNA. And so the place where transcription starts, right here and here on the RNA, is different from the place that that translation starts. This little intervening region is called the 5' untranslated region. Sometimes it's also called the leader sequence. Likewise, at the end, so the protein coding region will end, you'll have the stop codon, and then there's a little trailer sequence, as sometimes it's called, or the 3' untranslated region, which continues on until the place where transcription ended, which is right at the end of that RNA. So if you look at the genome, the genome sequence of an organism, right? How is it that the cell knows where the genes reside, right? It just looks like a whole big gibberish of G's, A's, T's, and C's, right? But there are particular sequences that the cell can recognize and say, aha, that's a gene. So one of those sequences is in the promoter region. And again, so let's just look at this figure, this diagram. So for every single gene, there's a spot where transcription will start, and that's called the +1 site. Okay, it's shown in this figure right here after the promoter. And then there's a little intervening sequence here where you'll get RNA. This is going to be the 5' UTR. And then this is the beginning of the protein coding region, the ATG, that codes for methionine. Now, if you look at a number of different genes, and here's a whole bunch of genes that we've lined up the sequence, all of them, their +1 sequence starting at the same place here. If you look in the promoter, which is before that +1 site, what you'll find is that there are certain conserved sequences that are found in all genes. Now, if you go backwards from the +1 site, we call these the minus, right? This would be -1, -2, etc. And about 10 bases before that +1 site is a region of conserved sequence called the -10 box, and that sequence is something like TAT-AAT. And then you go back another approximately 15 bases additional, and you get to another conserved sequence, which is at the -35 position. And so these two conserved sequences are actually where the RNA polymerase will bind, and help to recognize that there's a gene there, and the RNA polymerase should bind so transcription can happen. So those are the DNA sequences that are important, the -10 and the -35 sequences. Now let's talk about the proteins which bind to those sequences. So there are two main parts. There's something called the core enzyme of RNA polymerase, and this consists of two alpha subunits, a beta subunit, beta prime, and an omega subunit. There's also the sigma factor, which is shown in blue here. And so it's the sigma factor that actually binds to those conserved sequences in the promoter at the -10 and the -35 boxes. And the sigma factor binds there, and then it recruits the rest of that core enzyme, all of those other subunits. And once all of this stuff is sitting there ready to go, the sigma factor is what actually separates those two strands of DNA to allow transcription to begin. Once the sigma factor does that and transcription begins, the sigma actually falls off. It's completed its job, and it'll go on to bind to another promoter. In contrast, the rest of that core enzyme is going to continue on transcribing during elongation. One thing I just want to say is that bacteria actually have a couple of different versions of their sigma factors. So this one is called sigma 70. This is commonly used during normal vegetative growth, but there are some other sigma factors which are used in sort of unusual circumstances and have slightly different recognition sequences on the DNA sequence. Okay, so that core enzyme now is shown as this purple oval. And during elongation, what happens is the RNA will start to be formed, and you have these new ribonucleotides coming in, complementary base pairing with the template strand of the DNA, and about 10 nucleotides of DNA RNA duplex will form in this transcription bubble. And as this RNA gets longer, then what's going to happen is that extra RNA will start to kind of hang out the end of that transcription bubble. As transcription on this gene continues, that DNA is going to continue unwinding in this direction, so more ribonucleotides can be added to the 3' end here, and it will rewind in this direction. So you have a small region that's moving down the DNA and synthesizing a RNA as you go. The last stage of transcription is termination, and I just want to point out that elongation is going to continue past the end of the protein coding region on the RNA and into this 3' untranslated region till it finally hits that termination sequence. So in prokaryotes, there are two kinds of termination sequences, intrinsic and row-dependent. So we'll talk about intrinsic first, and this is called intrinsic because it is based solely on the sequence of the RNA and the DNA that is being transcribed, and there are no other factors that have to be involved in this process. So intrinsic termination depends on the formation of something called a hairpin loop in the RNA structure that is being transcribed. And so this RNA that has just been transcribed will actually base pair with itself and form this little loop with these base pairs that are a complementary base pairing with each other. Immediately following that hairpin is a series of U's on the RNA, okay, and it's A's in the DNA, shown up here. And what happens here is when there's a string of A's and U's, remember that these form only 2 hydrogen bonds in the base pairing between those, and it's a little bit weaker interaction than if there were some G's and C's in there. And so what happens is that the RNA polymerase kind of senses that this is a little destabilized, and it will pause and just try to backtrack a little bit in order to make sure that everything's okay. Now, it can't backtrack very well because that RNA is forming this hairpin loop. And so what happens in that case is once the RNA polymerase tries to backtrack, it just completely destabilizes, and the whole thing falls apart at that point. Okay, so the RNA comes loose, the RNA polymerase comes off, and the DNA winds back together again. The second type of termination signal is something it is called row-dependent termination, and it is called this because it uses a protein called Rho, and it's just shown in this figure in blue. And so here's the RNA that's coming out of that transcription bubble, and there is a particular sequence on the RNA that is being synthesized called the RUT site, R-U-T. And the RUT site is where Rho actually binds, and then Rho sort of somehow binds the RNA in a certain way that causes it to be destabilized. And so it kind of pulls that RNA out of that transcription bubble, and everything falls apart. Okay, so the RNA is free, and on this particular figure, it shows that it's already starting to be translated, and the RNA polymerase falls off, the DNA-DNA hybrid reforms.
Okay, now we're going to move on and see a video that differentiates row-dependent from row-independent. I know this previous video didn't explain a whole lot. So there are 2 classes of transcription terminators. They are DNA sequences. There's the row-dependent and the row-independent. The row-independent is also known as the intrinsic. And the sequences are on DNA, and they're responsible for triggering the end of transcription. So they're going to code for the 3' untranslated region. So it's important to remember that the stop codon of messenger RNA signals termination of translation, not the termination of transcription. Okay. So you can read here on row-dependent terminators and row-independent terminators on your slides. And I would like to show you a comparison of the 2. Let's see where they are. Oops. There it is. Couldn't see that there. All right.
In this video, I want to talk about how transcription ends, or how transcription terminates in prokaryotes specifically. So there are 2 ways. There is row-independent termination, which is also called intrinsic termination. And there's row-dependent termination. Rho is a protein, and we'll talk about it shortly. But in the case of row-independent termination, the Rho protein is not used. Instead, what happens is a hairpin loop forms due to these things called inverted repeats in the DNA, and that signals the end of transcription. So if we imagine this being our DNA strand, an RNA polymerase is going along, transcribing RNA, eventually this thing, the mRNA itself, loops around itself. This thing is the hairpin loop. Once that hairpin loop forms, that signals to the RNA polymerase to stop transcribing, and then the RNA polymerase falls off. Now this RNA polymerase is in a prokaryote, right? So it must be the core enzyme. So how does this hairpin loop actually form? That's something we'll get to in just a moment after we talk about an overview of row-dependent termination. So if it's row-dependent termination, the Rho protein is used. So what happens? The Rho protein, keep in mind this little this little symbol is the Greek symbol for rho. Rho travels along the RNA transcript until it reaches RNA polymerase. And then it causes the RNA polymerase to dissociate from the DNA. Okay, so if we imagine this to be our DNA strand, this is RNA polymerase, it's going through, making RNA, then this little Rho protein hops along. Now Rho is going to travel along this mRNA until it reaches RNA polymerase. And when it does, it's going to cause the RNA polymerase to fall off, dissociate, and of course the Rho protein itself will fall off. So that is how Rho-dependent termination works. That's pretty much it. Now let's talk a little bit more about Rho-independent termination, how this hairpin loop forms. We said that it forms due to these things called inverted repeats in DNA. So here are details on the inverted repeats. So if this DNA strand is running 5' to this, 5' to 3' this way, and 5' to 3' this way for the bottom strand, let's imagine this top strand being the template strand. So this here, this green portion is, or these green portions rather, are the portions in which we have these things called inverted repeats. So this is an inverted repeat here, inverted repeat, oops, repeat. And over here, this is also an inverted repeat. I'll just put inverted repeat IR here, okay? Why is that? What does that even mean? Well, notice that here we have G, 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 C, 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 G, C, G. Now, if we start off from here to here, right? Or actually, here we had 9 bases, right? 3 G's, 3 C's, and a G, C, G. Over here, we also have 9, and it just so happens that if you start here, it's going to be complementary to these. So here, we ended with a G. Here, we're starting with a C. So the C is complementary to this G. So if you read this one going this direction, you will find that it's complementary to these going in this direction. So here we have a C is complementary to the G. A G is complementary to the C. The C complementary to this G. These 3 G's complementary to these 3 C's, and these 3 C's complementary to these 3 G's. So what happens when this template strand is transcribed into RNA? Well, this is what we get. We get this RNA strand. Okay, this strand here is the RNA. And it has these inverted repeats that are bound by the red lines here and written in green. So what happens here is that this strand sort of bends on itself. This side comes in this way, and this side kind of bends in this way. And then we have the loop formation. And why is that? Well, because now this C, if I can get a good color here, if I can get this C, is going to hydrogen bond with this G. This G is going to hydrogen bond with this C. And this C will hydrogen bond with this G. And the rest of them are complementary, so they're all going to hydrogen bond. So we're going to have hydrogen bonds here, 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 and here. And that's going to hold this hairpin loop together. And typically something to note about this is that usually these are very GC-rich regions. Why would that make sense? Well, GC base pairs have 3 hydrogen bonds. AU base pairs have 2 hydrogen bonds. So if we want to make sure that this hairpin loop stays together, it would make sense that the GC base pairs kind of make up at least most of it. Here I've drawn it all. It's not necessarily going to be the case. I just wanted to emphasize the point or exaggerate the point. But there could be AU base pairs in here. So that's how this hairpin loop forms. And once this hairpin loop forms, it signals to the RNA polymerase, hey, stop polymerizing. We're done here. Okay? And those are the ways that termination can occur in prokaryotes. Now, what about termination of transcription in eukaryotes? It's a good question, and the reason why it's a good question is because this idea is not really totally understood yet. It's not really well understood how termination of transcription occurs in eukaryotes. So I'm not going to make a video about it simply because I don't know, and scientists don't really know too well. So I hope that video was helpful overall, but not disappointing at this little point here.
All right, so for assessment question number 4, you're going to go back and you're going to take a look at the 2 consensus sequences on that slide that you're going to find complementary at -35 and at -10. What's in the shaded box is just your choice. +1 is going to begin the leader sequence, and remember the leader sequence is going to encode the Shine-Dalgarno sequence. The Shine-Dalgarno sequence is shown in a previous slide, and remember that sequence is messenger RNA. Messenger RNA is synthesized in the 5' to 3' direction, so you have to work backwards to see what's going to be on the 3' strand and the 5' strand of DNA. And then remember the last 3 boxes encode the start codon. The start codon, remember, in messenger RNA is AUG. It's 5', AUG, 3'. So again, you have to work backwards to find out what the DNA sequence is going to be. Assessment question number 5 now. You are going to rewatch the videos, reread the information. You're going to tell me what's necessary for initiation. Those are the requirements. Make sure you include the enzymes and any sequences that are involved. What is the outcome in the next box? The outcome of each of these events. What are the products? And then the purpose is the last thing. And when you type in these boxes on your assessment, the boxes are going to accommodate your typing and get larger. Next up is going to be translation.